The nematodes pest feed on plants, reducing yield and growth in infected plants. Plant parasitic nematodes primarily affect the roots and induce general symptoms of root inadequacy such as yellowing of leaf, wilting, and thrifty growth and chlorosis. Plant pathogenic nematodes are marked by possessing a needle-like structure in the mouth referred to as stylet. Depending on the species of nematode, stylet is used to feed on plant pods which may include roots, leaves, seeds and stems. Plant parasitic nematodes may result in significant economic damage in nearly all the vegetable crop, fruit crops, field crops, fiber crops, plantation crops, pulses, oilseed, ornamental crops, etc. by reducing both quality of the produce and yield. Globally, the annual yield loss resulted by nematodes amounts to about $78 to $80 billion. Over a period of time, some nematodes symptoms could be accurately diagnosed in the field. But in order to confirm field observation, laboratory assay of soil and root is usually necessary. Soil and root sampling. Owing to their microscopic nature, nematodes cannot be observed with the naked eye. Over a period of time, this microscopic organism can build up to population levels and can result in significant economic damage. This includes yellowing, stunting, wilting, root abnormalities and reduced yield. But these tropical symptoms alone cannot ascertain nematode damage to plants. In order to correctly detect nematode infection, soil and root sampling of the infected field is mandatory. Root and soil samples have to be extracted and sent to laboratory so as to identify and count the nematode linked to the injured plants. Soil and root sampling enables one to diagnose the type of nematodes infecting the field and also helps in establishing the susceptibility of a proposed crop. This in turn helps in predicting the damage to the crop. The patterns of nematode distribution in the field may also be detected using sampling methods. Discovering the nematode distribution in the field may permit partial treatment, reducing the economic and environmental impacts of the methods used. Therefore, accurate diagnosis of nematode induced disease or injury generally requires soil laboratory analysis. The first step in the diagnosis of crop disorders caused by nematodes includes collection of soil samples and roots of the nematode infected plants. Proper collection and processing of the soil samples goes a long way in accurately diagnosing the nematodes. Inappropriate collection and handling of the soil samples may result in the dismissal of nematodes. The proper guidelines should be followed while collecting samples for sampling of nematodes. Time of sampling. Farmers and growers should take soil and root sample for nematode testing at a regular interval, especially just before planting and at the time of harvest. Time of nematode sampling is particularly important because the population of nematodes fluctuate throughout the year. They may be undetectable during the winter and early spring, but it often intensifies to a very high population density in the early fall before crop harvest when live roots are present. Immediately after harvest, nematode population may decline abruptly. Therefore, samples have to be taken for nematode testing at appropriate time. This in turn assists in avoiding the risk of failing to identify a damaging nematode species, thereby avoiding or reducing the probability of nematode problems in future. Also, sampling in the early fall eases for decision making in executing appropriate nematode management practices before the next crop is planted. Growers should maintain a record of population levels in different blocks in various blocks before planting. Reduction, if any, of nematode population due to shut-in treatment, etc., so as to assess the efficiency of a particular management programs. Also, it should be noted that nematode management practices are need-based and growers are suggested to treat the soil if only they have identified specific nematode species that causes damage to a particular crop. Sample collection. Laboratory determination is a vital tool for assessing nematode problems. Nonetheless, an analysis can only be as good as the samples. Therefore, proper sampling procedures are critical. Nematodes are unequally distributed in the soil, both vertically and horizontally. The distribution of plant parasitic nematodes in the field is random and no uniform. Their distribution in the field is irregular and is aggregated as clusters. Thus, determination of nematode population densities is naturally imprecise and may be subjected to a high degree of variability. Owing to patchy distribution of nematode, soil samples have to be collected from many spots around the field instead of collecting from one or two spots. This ensures that the soil samples are truly representative of the area sampled. The guideline of sampling. The upper 1 to 2 inches of soil layer is removed as nematodes are usually not present in that region, and then samples are taken using a 2.5 cm diameter soil probe. 20 to 30 soil cores in a zigzag pattern are taken from regularly spaced locations throughout a large area or a small area. Fruits and ornamentals may be sampled under the canopy drip line, and the number of soil cores taken depends on the size of the tree or a shrub canopy. The soil cores collected should be thoroughly mixed into one composite sample, and about a pint of soil should be transferred into a labeled plastic bag for nematode assay. Handling of soil samples. The soil samples are collected, the biological vitality of the sample should be preserved because the extraction and identification procedure requires live nematodes to attain the best accuracy. Once collected, the soil and root sample should be delivered to the diagnostic laboratory without any delay. It should be ensured that the soil samples should be protected from extreme of temperatures. Also, the samples should not be allowed to sit in direct sunlight in the field or for even a short time. In order to achieve this, samples should be packed in insulated containers and placed in cool environment. Once in the laboratory, samples can be stored in refrigerator until the nematodes are processed for extraction. Extracting nematodes from soil. Plant parasitic nematodes may be extracted from soil and infected plant materials in many different ways. No single technique will efficiently extract all sizes and kinds of nematodes, and therefore endless range of extraction tools and techniques have been developed for extraction of nematodes. Some are efficient for extraction of nematodes from soil, while the other work best for extraction from roots and seeds. Some others are specialized for nursery stock inspection. Some are partially used, others have been adapted to suit local conditions and requirements and availability of equipments. Some of the commonly employed nematode extraction methods are corpse sieving method. Carp sieving and gravity method is a swift method, take less than 30 minutes and permits the recovery of active sluggish and some dead nematodes from large soil samples. This procedure enables the recovery of eggs and chest as well. The sieves. Stainless steel sieves of accurately graded size are used. These sieves are quite expensive. It can be substituted with sieve made out of plastic pipe and nylon mesh cloth of known mesh size, which is comparatively cost effective. These are fixed over pipe like embroidery hoops. Also, plastic cups are cut across in half and nested together in order to construct sieves. The effectiveness of the sieve depends on the mesh number. Mesh number is the number of openings per inch of surface. For example, 105 or 625 openings per square inch can be found in a 325 mesh sieve. Procedure of this method. Start by thoroughly mixing the soil sample on a large sheet of paper or on a clean plastic. In a plastic bucket, place known volume of this mixed soil and about 1 to 1.5 liter of water. Gently mix the soil with water by continuous stirring in order to break up all clots. If the soil mixture is dry, ensure that the soil is choked for few hours. Continue stirring until a thick muddy mixture is obtained. Now, most of the nematodes are found suspended in water. Some workers make use of flocculating agent like Separon NP10 in order to break up soil aggregates and release nematode from the sample. Now, let this muddy mixture settle for 30 seconds to 1 minute. This is followed by decanting the muddy water through a 0 or 20 mesh sieve into the second bucket. This step removes trash and heavy soil particles. Care should be taken not to pour the sediment which is disposed later. Step 2 and step 3 have to be repeated for efficient recovery of nematodes, and after this most of these nematodes will be found in the second bucket. Now the nematodes in second bucket are suspended by stirring the water in the bucket. Then the water in the bucket is allowed to stand for a minute or two and the contents in the bucket is poured through a 150 or 200 mesh sieve into another clean bucket, avoiding the sediment. Next, a gentle stream of water is directed onto the upper side of the sieve at an angle of 35 to 40 degree, so to move the nematode to the edge of the screen. Water thus poured in is collected in a bucket. Small nematodes and eggs will be washed through the sieve and will be recovered later. Large nematodes are collected by backwashing, wherein a gentle stream of water is directed on the edge of the upper surface of the sieve and water is collected in a 250 milliliter beaker. Repeat these steps followed by backwashing into a 200 ml beaker. Now, resuspended small nematodes and eggs collected in that third bucket. Concentrate them on 325 mesh sieve and backwash them into 250 ml beaker. This is followed by concentrating the contents in the 250 ml by pouring through a 500 mesh sieve and backwashing with small quantity of water into a weaving dish or 50 ml beaker. Concentration of the samples can also be done by allowing the nematode to settle to the bottom of the beaker, followed by very carefully decanting all but the last 10 to 20 ml of water. Using this method, an experienced technician can recover a large percentage of nematode in a soil sample in about 20 minutes. The material thus collected from the sieves may be directly examined or it may be placed in Behrman funnel for additional separation of nematode from soil particle. Behrman funnel technique and modified Behrman funnel technique. Behrman funnel technique is the simplest method for extracting nematodes. This method is extremely cost effective, relatively quick, and yields live and active nematodes from soil and plant materials. But this technique is not quantitatively reliable as it selects against slow and non-moving nematodes. This technique makes use of three components: a large plastic tray, kitchen paper, and a wide mesh. The tray may be of the size used in dissection or may be of the size of photographic developing tray. The mesh required may be a single layer of thick plastic or rubber netting. The mesh used may also be a plastic coated metal grid covered with thin plastic couch. Bare metal mesh cannot be used because the metal may release toxins. Suitable mesh is placed in the tray and its sides are covered with a single sheet of kitchen paper. Then a thin uniform layer of soil is spread over the paper. Now clean water is poured gently into the tray until the soil is wet but not submerged. Spillage of soil particle along the side is avoided as they cloud up the extract and make it tedious in observe the nematodes. The setup is left undisturbed until the next day. Active nematodes crawls down through the kitchen paper and seep to the bottom of the tray. Thus, a large proportion of the sample nematodes will collect on the bottom of the tray overnight, ready for further treatment on the next day. Suspended nematodes are concentrated by pouring the water out of the tray through a fine sieve, followed by washing the nematodes off into a small beaker with about 20 to 30 ml of water. Alternatively, the extract water is poured into a sufficiently large beaker and the nematodes are allowed to settle. Later, topmost water layers are discarded and the remaining water is taken in a small beaker. This process is repeated until only 20 to 30 ml suspension is left, which now has thousands of nematodes ready for further processing. This method is referred to as active method and is not helpful for inactive nematodes like Longitorids, Trichodoris and Cricnonematidates. Thus, the technique has been modified and supplemented with other techniques so that it becomes the standard method for extraction of nematodes from soil and plant tissues. Modified Behrman funnel technique. This method makes use of a supporting mesh in order to hold the plant material partially immersed in water. This evades anaerobic decomposition. At the bottom of a plastic ring cut from polythene or vinyl tubes, a supporting couch mesh is fixed. Now, wet strength a facial tissue or milk filter and place it on the supporting mesh from which active nematodes can be extracted. Later, a thin layer of soil or chopper plant material are placed onto the tissue and water is added through the funnel so that the tissue is partially immersed in water. After about 24 to 48 hours, nematodes are collected. Sugar flotation technique. This method involves centrifugation of soil sample in the sugar solution which suspended the nematodes in the supernatant while the soil particles get deposited at the bottom. About 20 grams of soil samples was taken in each of the 6 plastic centrifuge tubes and water was added to it within 1 centimeter of the lip. With the help of metal spatula, the soil and water in the tube is mixed well, followed by addition of a liquid of nematode, and the suspension was remixed before centrifugation. After centrifugation, the supernatant was decanted and, like in previous step, sugar solution was added and mixed. Again the sugar suspension was centrifuged at 700 rpm for 1 minute. After the second round of the supernatant was decanted and passed through a 38 dm sieve, and the nematodes are collected by backwashing from the sieve and counted. Plant parasitic nematodes are microscopic in nature and cannot be observed with naked eyes. Over a period of time, these nematode pests feed on plants, reducing yield and growth in infected plants, and can build up to population levels which can result in significant economic damage. Nematode infected plants show symptoms such as yellowing, stunting, wilting, root abnormalities, and reduced yield. But these tropical symptoms alone cannot ascertain nematode damage to plants. However, in order to correctly diagnose nematode infection, soil and root sampling of the infected field is mandatory. Root and soil samples have to be excited and sent to laboratory for identification and further counting of the nematodes linked to the injured plants. No single technique is efficient in extracting all kinds and sizes of nematodes. Therefore, an endless range of extraction tools and techniques have been developed for efficient extraction. Here some simple, widely used and efficient methods of extraction like corpse sieving method, Behrman funnel technique and modified Behrman funnel technique and sugar plantation technique have been discussed in today's session.